Okay, can you see my presentation?
Yes.
Okay. Good morning, everyone, and thank you, Professor Reyes, for inviting me to join this very interesting webinar. I'm a researcher at the chemistry department of Università della Calabria, and I'm working in the mass spectrometry lab of this department. Today, I'm going to talk about one of my main scientific interests, the quality and safety of olive oil and the development of new innovative mass spectrometric approaches for the assessment of these nutraceutical properties of olive oil. Olive oil is an important ingredient of our diet. It is considered a functional food because of its nutraceutical properties and beneficial effects on the human health. And these properties are related to its chemistry, to its particular chemical composition. It is a fat matrix, but it contains also low amount of several antioxidant molecules, such as phenols, carotenoids, and vitamins. But how can we assess the quality and safety of this product? Mass spectrometry is the most useful tool to address this task. Because it allows to obtain very sensitive and specific results in terms of identification, structural characterization, and also quantitative determination. We can evaluate the amount of specific quality markers, or we can perform MS fingerprinting analysis, MS profiling analysis to characterize the matrix or to characterize a specific class of molecules. Mass spectrometry has rapidly evolved during the past decades, especially with the development of the new class of ionization methodologies, the ambient mass spectrometry. And these techniques allow rapid and direct analysis of a wide range of samples of matrix. The ions are generated outside the instrument and at ambient condition after the desorption of molecules from condensed phase. And these techniques allow to perform analysis of samples in their native states, and so without or with a minimal simple pretreatment. Among these ambient ionization methods, the most useful are DAISY, PS, DART, or LTP MS. As regard of olive oil, the quality of olive oil, many approaches were developed for the assessment of quality and safety of these metrics. Some of these are based on the characterization of metrics. So by performing MS fingerprinting of MS profiling of triacylglycerols fraction or fatty acid fraction or phenolic compounds. And some of these procedures are based also on the quantification of specific quality marker, and so phenolic compounds, tocopherol, and sterols. In our lab, we have built an open source for paper spray application using a triple quadruple, originally equipped with an ESI source, which was replaced with an in-house implemented open source for PSMS application. It is composed by a high voltage clip and metallic stage for supporting. As we know, the paper spray experiments are very simple and cheap. A few microliters of sample of solution were spotted onto a paper triangle positioned in front of the MS inlet, and the ions are generated by applying a high voltage to the paper and a small volume of polar solvents such as methanol or water methanol. And the electric field generated between the paper and the MS inlet generate an accumulation of charges at the apex of the paper, and a spray, a charged spray. And after the solvent evaporation, the gas phase ions are analyzed by MS instrument. The research activity of my research group is focused on the development of new method based on mass spectrometry to assess the quality of a wide range of food, and in order to valorize our local resource. But today I'm going to present you our latest work, the quantitative determination of phenolic compounds and the quantitative determination of vitamin E in extra virgin olive oil. And now we are working also to the application of paper spray mass spectrometry for the characterization of vegetable oils by performing tags profiling in order to discriminate extra virgin olive oil from other vegetable oils which are commonly used as adulterants. The first work deals with the absolute determination of hydroxytyrosol, tyrosol, and their derivatives by paper spray, tandem mass spectrometry, and isotope dilution method. The family of phenolic compounds present in extra virgin olive oil is composed by the two simple alcohols, hydroxytyrosol and tyrosol, and their ester derivatives. The idea of this work born to address a recent regulation, a recent European regulation, which establish a list of permitted health claims made on food. In the case of olive oil polyphenols, this regulation permit this health claim. Olive oil polyphenols contribute to the protection of blood lipids from oxidative stress. But this claim can be used only for an oil which contains at least 5 milligrams of this compound per 20 grams of oil. The problem is that to date there is not an official analytical method to quantify these compounds. The International Olive Council recommend a classical HPLC UV analysis after an extraction procedures of the sample. But this procedure is not accurate because it lacks of suitable internal standards and because of the stability of the derivatives of tyrosol and hydroxytyrosol. This molecule, in fact, are dialdehydic species, and in the presence of solvents such as water and methanol, it can produce hemiacetal and acetal species, and this complicate the UV profile and so the results. We have previously developed a new method to obtain only hydroxytyrosol and tyrosol in order to quantify the total amount of hydroxytyrosol and tyrosol after a fast microwave assisted hydrolysis. And this is the experimental approach. After the adding of labeled internal standard, we can quantify the free fraction of the two simple alcohols and the total fraction of tyrosol and hydroxytyrosol after a fast extraction and a fast microwave hydrolysis for 4 minutes. After fast purification steps, the reaction products are directly analyzed by PSMS analysis for 2 minutes of acquisition time. The MS determination, the quantitative determination, was performed under MRAM condition and isotope dilution method. And this is the ion current of monitored transition for hydroxytyrosol and its internal standard containing 2 deuterium. And this current is used for quantitative determination. The obtained results, limit of detection, quantification, and accuracy are quite good. The accuracy in all cases is around 100%. This research work deals with the evaluation of vitamin E, also in this case by paper spray, tandem mass spectrometry, and isotope dilution method. And also in this case, this work served this recent European regulation, which in the case of vitamin E, it permits this health claim. Vitamin E contributes to the protection of cells from oxidative stress, and this health claim can be used only for an oil which contains at least 90 milligram per kilogram of oil. Vitamin E is a pool of antioxidant molecule, but in extra virgin olive oil, the isoform alpha constitutes about 90% of the total amount of tocopherols. And so we have decided to quantify this isoform. In this case, the experimental approach is very fast because we can say that the oil is directly analyzed by PSMS analysis, because after the adding of internal standard, also in this case labeled internal standard alpha tocopherol containing 6-deuterium, the sample was dissolved in acetone and directly spotted onto the paper. The quantitative determination was performed under MRM condition, and also in this case the obtained results are quite good, accuracy and limit of detection and quantification. Last but not least, I want to introduce you our recent research work. This work deals with the application of pepper spray mass spectrometry to characterize the triacylglycerols profile of different kind of vegetable oils, because tax profile can represent a fingerprint of the vegetable oils, and this MS profile was used coupled with statistical analysis in order to discriminate extra virgin olive oil from other vegetable oils used as adulterants. This is the experimental approach, and each vegetable oil were mixed with ammonium acetate, and after the dissolution in acetone, the sample were directly analyzed by PSMS experiment. This is the mass spectra relative to tax profile of extra virgin olive oil. Well, these are the the tax profile of other vegetable oils: sunflower, soybean, corn, and peanut oil. Several MS in paper spray and ionization mode were performed to characterize the most important ions present in mass spectra. And finally, we have used the tax MS profile to perform statistical analysis. And in this slide, there are the LDA score plot in this first figure of vegetable oils, and as you can see, the extra virgin olive oil cluster is well discriminated from other cluster which represent the other vegetable oils. And finally, we have prepared also the mixture of oils. And also in this case, the cluster of pure olive oil is well discriminated from the other mixture. In conclusion, we can say that this work, these presented works, confirm the ability of paper spray mass spectrometry to perform very fast quantitative analysis, but with the use of isotope dilution method, it allowed to obtain accurate results, and the ability of this technique to obtain fast molecular screening, fast mass profile, to characterize also a complex matrix like olive oil. Thank you for your attention.
Thank you very much, Lucia. It was a really interesting application because it's an actual application in which we are using ambient mass spectrometry to try to fix or to meet with a criteria set by regulation, in this case by European Union. So this is really important to show that this is not a toy, but something that can be really useful for laboratories. Anyone of you of the audience have any question?
Hi, Juan, can I?
Yeah, sure. Marcos?
So hi, Lucia. Really nice presentation. I just want to ask you, they are really two silly questions, but the tyrosols and the alpha tocopherol, did you measure it in positive or negative ion mode?
Sorry.
He is asking you the ionization mode, whether it is positive or negative.
For alpha tocopherol?
Yep.
In negative ion mode.
And for the tyrosol in positive or negative? Because you use HCl.
For hydroxytyrosol?
Yeah.
Negative ion mode.
Okay, both. Okay. Even when you use for the extraction HCl?
Can you repeat, please?
HCl is hydrochloric acid, this hydrolysis, the hydrolysis step.
Yes.
It's not a problem to analyze that in negative ion mode?
No.
Okay.
No. Yes.
And the other one is, I know that you corrected probably with internal standard, but why the paper spray peaks look like or that picky? Like you have peaks?
Sorry, but I can't.
So the previous slide. So the one that you have now, the previous slide, for example, you have the TIC for the paper spray analysis, and it looks like you have peaks while you're measuring.
Sorry, but I don't understand. I think that I have some issue.
Marcos referred to the total ion current signals. He thinks that they are really like dirty or
not dirty. It's like I assume that you could have like a flat line, or it was what I was expecting, and it's like peaks.
So, yes, this is simply an ion current of monitored transition. Mm-hmm. And we used the area of averaged over the total acquisition time for the quantitative determination.
Okay.
Yeah, it's not. This is common because the paper spray signal is not short, so it depends on the condition.
Yes,
it can vary from a few seconds to over 1 minute. So,
yeah. Okay.
2 minutes. Okay, only 2 minutes.
Yeah. You need, depending on the volume you spot on the paper, it can be longer, or it depends on many aspects. Probably, I guess.
We spotted 15 microliters of solution.
This is quite a lot. Probably, if you spot maybe 2 microliters, then the signals are really.
It should be just big.
Yes.
But then you may have the problem of that the sample may be stick to the the paper and you won't see it. So this is something you need to optimize for each particular application, I guess.
Okay, thank you very much.
So any more questions? Thank you, Lucia. Any more questions for Lucia? Bienvenida. Has the...
Yes, I have a couple of questions.
Go ahead.
Well, first of all, thanks for your presentation. It's very interesting.
Thank you.
The first thing I want to ask you, maybe, I don't know if you have said that during the presentation. Sorry if you have explained it before. How do you prepare the paper triangle? So do you do it by hand, or do you have like an established method to make it reproducible?
No, the paper, I prepared the paper using Batman paper, classical Batman paper.
Okay, because yeah, sometimes if I've seen that you have a very nice reproducibility, and always the cut of the paper influences the spray. So maybe this also influences your signal.
Yes, also the position of the triangle paper influence the ionization efficiency, for example, and the reproducibility of the experiments. Yes.
Have you tried different papers? I mean, the different composition or pore size, and have you noticed that you have, for example, adsorbents of some analytes in any kind of paper? Do you have some information about this?
No, at the moment, I don't try other kind of paper.
And sorry, I have just one last question. In your last work,
yes,
which is the minimum percentage of adulterant that you could detect to discriminate between real olive oil and adulterated olive oil?
Okay, so at the moment we have tried only a mix, 80% of olive oil and 20% of 20% of other vegetable oils. But yes, I will try to perform other analysis with different mixture. These are preliminary results.
Okay, yeah. I don't think it is so bad. So 20%, it's okay. I think it's better if you try to discriminate them between the fatty acid profile.
Yes, tax profile. Yes.
Yeah. So thank you very much.
Thank you.
Any more questions?
I have also a question.
Odysseus, go ahead.
Congratulations for the presentation.
Thank you.
I have a question about the linear discriminant analysis. As I see, your spectra is normalized at 100%, and the shape is similar in the most of the cases.
Yes.
But I can understand why you have a diversity in the black points. I can see the olive oil 80%. I think the label is. So yes. This diversity of the points has to do with characteristics of the olive oil, or is something as error from the data?
Not is the linear discriminant analysis was based on the total data recorded in the mass range of triacylglycerols ions.
Okay.
So Odysseus, what you use is a lot of signals, and I think these dots are not just signals, but they can be ratios of signals, or this type of data, treatment data is beyond the individual mass spectral signals, right? Am I right, Lucia? So,
yes, we have used the total ion current in this all of the ions present in the mass spectra.
Okay.
But you cannot associate a dot with a particular signal, a particular mass, right?
Yes, the software has retained only few variable relative to the ions. Yes. But these signals are referred at the triacylglycerols. Okay.
Okay, thank you.
Thank you very much so far for the attention. I hope that the webinar is interesting. As you know, we have